Okay, so in this video we're going to look at blood typing. And so blood type refers to blood group, right? And so we say that your red blood cells can have these glycoprotein spikes in their plasma membrane, right? And so these glycoprotein spikes are essentially like a name tag of the cell. And so when we talk about, well, what types of glycoprotein spikes are present, that refers to your blood type. And another word for those glycoprotein spikes we can say is antigen, right? So an antigen is just a molecule you're going to find on the surface of the red blood cell, and it's something that your immune system can use to recognize, self versus non-self, or like a cell that should exist in your body versus like a foreign cell, right? And so it turns out we have 30 different types of glycoprotein antigens on our red blood cells, but there's really only about 3 that we talk about with respect to blood type, right? We talk about A, B, and D antigens, but there are 27 other different types of antigens for blood type. So the reason why we mostly focus on A, B, and D is that those are the ones that are more immunogenic. They kind of have the strongest immune reactions if you have a transfusion reaction. So that's usually what we talk about. But what this means, though, you guys, is that if somebody says they have the same blood type as you, right? They're like, oh yeah, I'm also AB positive, right? Well, that's only 3 of the 30 different types of glycoprotein antigens. So even though you think you might have a similar blood type, it may actually be different enough to where it wouldn't be a good transfusion. And so it's more complex than maybe what you've heard before. However, we'll still focus on just those 3 for this portion of the lecture. So the reason why we talk about this, you guys, is that if you have a mismatch of blood typing, you can get an agglutination reaction, which is a clumping reaction. And what happens here is that the antibodies that you might find floating around in the blood plasma, they can stick on to foreign antigens. And when they stick, they cause those cells to kind of clump up or agglutinate. And so what you want to do for proper blood transfusions is to prevent this agglutination. And the way you do that is make sure that you're not introducing foreign antigens into someone's body, so that you're only giving someone blood that they're either not going to react to, or they're already antigens they have in their body. So for the blood groups, we have A, B, AB, and O, right? So with A antigen, that's just a specific shape of a glycoprotein molecule on the surface of the red blood cell. Same with B antigen, just a specific shape. So they're similar but different enough to have different letters, we say, right? And then AB would just be having A and B antigens present. And then O is not an antigen. That's important to know. So for O, even though it's another letter, O just means the absence of A or B, right? So there is no O antigen. It's just the absence of A and B. And so if you say you have type O blood, what you're saying is you're not saying that you have O antigen. You're just saying that you don't have A or B. That's what O means. So O is more of like a placeholder here, and it just means the absence of A and B. Okay. And so what we find, then, you guys, is that inside of our bodies, whatever your blood type is, you can make preformed antibodies against a foreign antigen. So let's say if your blood type were B, you're going to make anti-A antibodies, because A is something that you don't normally find in your body, right? Because if your blood type's only B, you don't normally have A inside your body, you're going to make anti-A antibodies. So if you're ever exposed to A antigen, what that's going to cause then is a agglutination reaction or clumping reaction, where your own body's antibodies are going to stick onto that A antigen, which is now foreign inside your bloodstream, right? And it causes that clumping. And you might wonder, well, why is that bad? Like, why do you care if your blood clumps up, right? Well, those tiny little clots, you guys, first of all, that can stimulate an immune reaction. And so you start getting things like excessive inflammation, which can lead to shock, okay? Also, those tiny little clots can clog your organs, especially organs that are, very well vascularized, like your lungs and your kidneys. So you can start to see organ failure from this. This is why, we're so worried about, having the proper blood types that are being introduced. So what we find, you guys, is that up until about 2 months of age, you're actually not making many antibodies. So you're saying like fetuses and newborns don't make these antibodies as much. And that kind of makes sense, because if the fetus was making antibodies and the fetus's blood type didn't match the mother's blood type, then you could start to get some weird reactions there, right? Where the fetal antibodies could start to attack the mother's blood, and that would be bad. So the way this works, you guys, is we're saying that up until about 2 months of age, as a fetus, you're not making anti-A or anti-B antibodies. And that's a way, kind of like a way for the fetus to avoid problems in utero because there's mixing of mother and fetal blood. Okay, so you see adult levels of antibodies by 8 and 10. So you're going to see that as the child gets a little older, right, they're going to be a little bit more sensitive to these types of transfusion reactions. But for a very young child, right, even a newborn, they're not going to be as sensitive to transfusions because they don't really have that many antibodies yet, right? So it's kind of interesting. So what this slide shows you guys are our blood groups. So AB, B, A, and O. If you have type O blood, which antigens do you have? Neither A or B. Very good. Right. What if you have type A blood? What antigens do you have? Just A. How about if you have type B? Just B. How about A and B? There you go. A, B. Good. That's what's shown here, guys. So what this means is that it's a surface antigen on the red blood cell, right? So this picture is showing A as being a little purple spike and B as being a green circle, right? And so they wouldn't really look like that in your red blood cells, but it's just a way to make them look different here. And so you're saying if you have type AB blood, would you normally make anti-A or anti-B antibodies? No, exactly, because that would mean you're attacking your own blood cells. So if you have type AB blood, you're not making anti-A or anti-B. So if you had type AB blood, could you receive blood from someone who had type B blood? Yeah, absolutely, because that's not different. You already have B in your body, right? What if you have type AB blood? Could you receive blood from someone who's type A? Absolutely. A is not different. You have A, right? So it doesn't have to be a perfect match. Like people who are AB don't need to have blood from people who are also only AB. Because if you have type AB blood, you can receive blood from someone who has only A or only B. Because neither of those things is different than what you already have in your body. And the whole point of this is you don't want your own body's antibodies to attack that new blood that's maybe going in your body, right? So with type B blood, we have B surface antigens, but would they make anti-A antibodies if you had type B blood? Yeah, because A is something you don't have, right? And so if you're type B and you're not exposed to, you don't normally have A in your body, then A is a foreign antigen. So your immune cells are going to make anti-A antibodies against A antigen. So if you're type B, can you receive blood from someone who has A in their blood? No. So if you're type B blood, could you receive blood from someone who had type AB? No, and here's why. Although someone who has type B blood could receive, maybe you would say that, okay, yeah, they have B in their AB, but the A in their AB blood type is still considered foreign. So you'd still get a clumping reaction, even though there's one similar attribute, because it's different by one antigen. That one difference of antigen is going to still cause a clumping reaction because if you have type B blood, you're going to be sensitive to this A antigen because you make anti-A antibodies. Good. How about if you have type A blood, you guys? What type of antibodies are you going to create? Anti-B. Very good. So if you have type A blood, could you receive blood from someone who had type AB blood? No, because the B would react, right? Could you receive blood from someone who had type O blood? Yes, because remember, O is not an antigen. O is the absence of A or B. So that's when we talk about O as being a universal donor, right? Because it lacks A or B, which means it can go to anybody who has A, B, or AB, right? Or even other O's, okay? So what about if you're type O, though? So I think about type O as being the most kind of altruistic, like it's the most giving, right? But it's also kind of bad if you have type O and you need blood from somebody else, right? Because if you have type O blood, you don't have A or B antigens on your red blood cell surface, so that means you're going to make anti-A and anti-B antibodies. So if you have type O blood, could you receive blood from someone who has type A? How about type B? How about type AB? No, exactly. Because all of those have foreign antigens according to your immune system, right? And so who could you receive blood from then if you're type O? Other O's. Very good. Now in the US, that can be more rare. However, blood types vary around the world. Like in Peru, O is the most common blood type. In the US, it's the second least abundant, right? You're saying it's the second most uncommon. And so it depends where you are. And the reason why these things can vary worldwide, you guys, is that your blood type's based on your genetics. And since genetics vary around the world, then the blood type distributions vary around the world as well. Good. Okay. Oh yeah, here we go. So this shows frequency and percent of population. I think it's kind of interesting how they broke it up like white, black, Asian, Native American. You know what I mean? Like, there's actually not a very strong correlation between genotype and skin color. So I think it's kind of interesting that the book broke it up like this. But hey, whatever. You know, that's how they did it. So what about Rh, you guys? Now, Rh is another type of antigen. We think about Rh as being rhesus factor. There's 52 different types of Rh. The most common one is D antigen, though, right? And so, sorry, not the most common, but it's the one it's D antigen is the one that indicates the positive sign, right? But the reason why we talk about this is that it's one of the most immunogenic, right? Which means that if you have D antigen, you wouldn't want to be donating blood to somebody who doesn't have D antigen because that can stimulate a very strong immune reaction, okay? Now, there are other types of antigens, though, for Rh, like you have C, D, and E, right? Those are pretty common. But there's 52 different types. And it's called Rh, you guys, because of rhesus factor, because rhesus monkeys have this as well, which is kind of interesting. So you're saying that 85% of Americans, right, are Rh positive. So 85% of people that live in the U.S., their blood type has Rh factor in it, right? So we say it's Rh positive or it has D antigen, rather. Okay. So it's pretty abundant. But we're saying only 15% have a negative blood type, right? They're lacking that. Now, here's the thing about Rh, you guys, is you don't make preformed anti-Rh antibodies if you have a negative blood type. Like if you're AB negative, you don't make anti-Rh antibodies unless you've been exposed before. So for someone who has a negative blood type, they could potentially receive blood from someone who has Rh once. But because their immune system would learn to recognize that foreign antigen, they would then create antibodies against Rh, which means they couldn't receive that a second time. Okay? This becomes a problem for pregnant mothers that maybe have Rh negative blood, but have an Rh positive fetus, right? Because some of those fetal blood cells can cross the placenta, and if those fetal blood cells cross the placenta and enter the mother's bloodstream, now you're saying that her immune system can start to recognize a foreign antigen, but it's the fetus's blood. And that type of condition we call is erythroblastosis fetalis. You guys will learn more about that in pathophysiology, but it's essentially a condition where the mother's immune system can start to attack fetal blood cells. But here's the thing too, guys, is it's not so severe on the first pregnancy. There's a higher risk of complications from this among the subsequent pregnancies. That kind of makes sense though, because if you have Rh negative blood type, you're not going to make antibodies right away. It takes time for your immune system to learn to identify this Rh antigen as being foreign. And so usually the first pregnancy isn't a problem. But you're talking about multiple pregnancies, right? Then you're saying that the mother might have preformed anti-Rh antibodies in her bloodstream. Some of those can cross the placenta, now get into the fetus's blood, and start to attack the fetal blood cells. And that could be life-threatening for the fetus and the mother. So it's kind of interesting. All right, guys. So in terms of testing blood type, and this is something we did in lab, right, is we actually added some serum, or basically like little samples of blood, to wells, right? So you're saying that all of these wells would just contain a sample of blood. All these wells contain a sample of blood as well. And then we added antibodies to our blood to see if it would clump or not, right? So if you added anti-A antibodies to this sample of blood and it clumped, what does that tell us? If you add anti-A antibodies to the blood and the blood all of a sudden clumps up, you're right, A is present. Because the blood won't clump when you add the antibodies unless it has something to stick to, right? Because it's the antibodies that cause the cells to kind of clump up. But if they have nothing to stick to, there won't be clumping, right? And so what we're saying here then is that if you see a clumping reaction like here, after adding a certain antibody, that means that antigen is present. You're just testing for the presence of that antigen, right? How about over here, you guys? If you add anti-B antibodies to this sample of blood and it clumps, what does that tell us? That B is present, right? So you can take both of these results together and say, oh yeah, they have type AB blood because clumping occurred. Good. How about down here, you guys? We saw clumping in anti-A, but no clumping in B. So what blood type's that? A, good. How about down here, you guys? No clumping when you add anti-A antibodies or anti-B antibodies. O, right? Neither A or B are present, because if no clumping reaction occurs, then that says that basically you don't have A or you don't have B. This is missing Rh though, right? You could also test for Rh factor by adding like anti-D antibodies, and you can have a third well going down here to know whether you're positive or negative, that kind of thing.